Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in the course Enzyme Science and Technology, we are discussing about the different properties of the enzymes and its contribution in the development of science as well as the technology. So far what we have discussed about the enzyme classifications and enzyme nomenclature, and in the previous module we have also discussed the history of the development of the field of enzymology. And in the current module we are discussing about the structure of the enzyme, and when we were discussing about the structure of enzyme, we have discussed about the primary structures. We have also discussed about how to determine the primary structures, and then subsequent to that we have also discussed about the secondary structure. We discussed about the tertiary structure, right? And when we were discussing about the secondary structure, we discussed about the how to determine the secondary structure with the help of the CD spectroscopy and as well as the IR spectroscopy. And in the previous lecture, we have also discussed about the different methods to determine the tertiary structures, and in that context we have discussed about the X-ray crystallography and as well as the NMR spectroscopy. So in the today's lecture we are going to discuss about the computational methods to determine the protein structures. So let's start today's lecture. So what we are discussing in this particular module is we are discussing about the protein structures. So as you can see that protein structure is made up of the, or protein structure is being presented by the four stages or four levels, right? So protein structure is being represented by the four levels: primary structure, secondary structure, tertiary structure, and some cases you also have the quaternary structures. Primary structure is the amino acid sequence of the proteins. Secondary structure is the alpha helix, beta sheets and turns, and all these secondary structure, when they come together and they will fold further, they are going to give rise to the tertiary structures. So while we were discussing about the structure determination of the tertiary structures in the previous module, we have discussed about the different methods, different experimental methods. So what are different experimental methods we have discussed? So in the experimental method we have discussed about the X-ray crystallography and as well as the NMR spectroscopy, and both of these methods are actually going to give us the protein structures. The structure what you are going to get from the X-ray crystallography is going to be a static structure, or and whereas in the case of NMR spectroscopy it is going to give you a dynamic structure or solution structure. So this is also called as the solution structures. Now in today's lecture we are going to discuss about the non-experimental method. So when you have a protein for which you would like to determine the protein structure, you have the two options: experimental procedures or the non-experimental procedure. So when we say non-experimental procedure, that means we are actually going to talk about the computational approach, what you can actually be able to use to determine a protein structure. So all these are actually being based on the determining or based on the data sets, right, which actually is going to be used to train the particular software, and that's how you are actually going to get the protein structures. So what we are discussing about is the molecular modeling of the protein structures using the computational tools. So when we talk about the molecular modeling can be of two types, okay, depending on the type of protein sequence what you are using. It could be ab initio or molecular modeling, or it could be the template based homology modeling. Under what condition you will do the ab initio or the initio molecular modeling? So when the protein is having no non-homologous sequences, or protein is going to have the or not available for the non-homologous protein structure, so it doesn't have any homologous structure, then you are no option but you are going to do the ab initio or molecular modeling. These are the places where you are actually working with the new proteins or new protein fold, and in that case it does not have the any kind of homology, and that actually is going to be very difficult to solve the get the protein structures. What are the things basically people do when they do the ab initio molecular modeling is they are actually going to use uh, the help of the protein folding. So, they are actually going to predict how the protein folding is going to occur if this is the protein sequence. So, they will looking at the protein sequence, and then looking at the protein sequence they will actually going to predict what will be the protein folding, which means what region of the protein is going to adopt what conformation, and then they will actually going to cross verify by that with the help of the Ramachandran plot and all other kinds of energy parameters. Because if you are actually going to get a good inner model, it is actually going to show you the low free energy, right. So that the molecule is going to be more and more stable. But if there are steric hindrance, if there are actually clashes between the side chains, that is actually going to be you will get that information from the Ramachandran plot, and that also is going to be reflected in terms of the energy parameters. So both of these parameters can be used to determine whether the predicted protein fold can be correct or wrong, and using this you can be able to go with the ab initio molecular modeling. Apart from this, they are also going to use the dynamics. So they are also going to unfold the protein and then again put it under the molecular dynamics stimulations, and that is how they are actually going to use. These are the three robust tools to predict the protein folds, and once they predict the protein fold, eventually they are actually going to get the 3D structures. So in today's lecture we are not going to discuss about the ab initio molecular modeling, because that is not very popular, because it requires the extensive computational tools, and you can imagine that if you have a protein of 100 amino acids, it is very difficult to predict the protein folds, especially when it is not having the homologous sequences and it is actually a new protein fold. So in that case you might have to generate the protein folds using the Ramachandran plot, and then you might have to determine what will be the free energy, because ultimately the free energy of the system has to be lower down while it is going through the protein folding process. Then the second approach is the molecular modeling. So molecular modeling, under what condition you will do the molecular modeling? So when you are working with a protein sequence which has homologous structures in the database, that time you will use the homology modeling, or so homology modeling is a multi-step process, and it is actually going to going to use the different process, and you can be able to use the different types of softwares to do the molecular modeling. Then what we are going to do is we are going to first discuss about how the molecular modeling is homology modeling is theoretically working, and then we are actually going to show you a small demo. So what is the molecular modeling or homology modeling? So this is a useful and fast structural solution method where the sequence similarity between the template and the target enzyme is used to model the three-dimensional structure of the target enzyme. The homology modeling exploits the idea that the amino acid sequence of a protein directs the folding of the molecule to adopt a suitable three-dimensional conformation within the minimum free energy. So what is the basic idea of doing the homology modeling? Homology modeling means that you are actually going to have the template structure, right? So template structure is actually going to give you two information. One, it is actually going to give you the information about the protein sequence, which actually it is going to be, right, which is from the protein sequence from the template protein, right, and it is also going to give you the information about the protein folding, right, how a particular sequence is actually getting folds into that particular thing. This means once you know the protein folding, you are actually going to get the 3D coordinates of the template residues, right. So so basically it is actually going to give you the XYZ coordinates of the template residues, how the alanine, glycine, arginine, everything is present in this particular template structure, right. Now, if we have the target protein, or if I have a target enzyme for which I am going to determine the structure, what I require is, or what information I have is actually the protein sequence from the target enzyme. Now, what I will do is I am going to take the 3D coordinates of the template residues, okay, or I will say the backbone, because the residues are actually going to be same or different in some cases. So, since both of these, the template structure and as well as target structures, are homologous, the many places the 3D coordinates of the template residues are actually going to be the same as it was present in the target enzyme, but many places or few places the template residues are going to be different, and because of that you are actually going to only take the 3D coordinates information of the template backbone, which means you are only going to take the peptide bond information. You are going to only the peptide bond or the main chain. On side chain information you will not going to take, because the side chain information may or may be same or may be different. So, this is what is exactly what you have to do, but if you want to do this, you have to follow the multiple steps so that you can be able to achieve this target. So, you cannot just simply take the 3D coordinates, you have to first determine and identify the templates, and that is why this is a homology modeling is a multi-step process. So, what are the steps? In the step 1, you are going to find the query sequence, right. So, you are actually going to have to identify or isolate the query sequence. So, query sequence is nothing but the amino acid sequence of the test enzyme. This you have to do from the NCBI server, right. So, you can actually if you know the protein's name or if you know the accession number, you can be able to get the query sequence from the NCBI server, right. Then once you got the sequence, then you are actually going to select the templates, right, because and that you are going to do with the help of the BLAST. So, what you are going to do is you are going to take this amino acid sequence and you are going to put it into the NCBI BLAST, which is a program actually, and NCBI BLAST, once you put it and you will select the database as the protein structure database, right, or the PDB. So, when you do the PDB database, it is actually going to give you the templates. So, it is only going to tell you that okay, these are the templates through which your amino acid sequence is matching. So, these are the potential templates, and then after that you are actually going to test these templates in the step 3. So, in the step 3 you are actually going to see the utility of each template, right. So, that you are going to do by a pairwise alignment of the query with the template. So, you are going to do like template 1 you are going to do like template 1 versus your test sequence, right? So you are going to do a pairwise cluster W, and you are going to use the program which is called as cluster W. So when you do the class that, it will actually going to tell you wherever you have the gaps and how much is actually it is having the identity. If you have a very high number of gaps, or if you have a very small identity, then it is actually not be suitable. So in that case you will reject the template 1. These are the things which can also be determined even by the some other kinds of scores. So after once you select the template, then you are going to use that template into the software, and it is actually going to allow you to build the model. So it is actually going to give you the 3D model, and it is actually going to do exactly the same phenomena, right? It is going to take the amino acid sequence from the target enzyme, and it is going to take the 3D coordinates from the your template structures, and then it is actually going to put them together, and that's how it is going to do the final refinements, and it will give you the 3D homology model. So it is going to be called as homology model, right? Once you have prepared the model, then it has to be validated with the help of the validation programs like Ramchandran plot or Verify 3D or the errata plot, right? So all these are you're going to use, and there are servers which are available to do this job. Okay, so I have given you a reference for this particular steps, so you can actually be able to go through with this reference and the title this article, and it is very good for determining or understanding the different steps. So far what we have discussed the theoretical aspects of how to determine or how to perform the homology modeling. Now what I'll do is I'll take you to my lab where students have prepared a small demo clip, and they will be going to show you the different steps.
In this video, I will be talking about the steps involved in the homology modeling using modular, and parallelly I will be performing it on screen and explaining each step in detail. So in step 1 we need to find out our query sequence which we need to model. So that query sequence, either we have the sequence from the literature, or we can download our query sequence from NCBI website given here. And then in the downloaded file we need to make some changes to make it compatible uh, for working with modular. So I will show you how to download and what changes we need to make. So we will open up the NCBI website, and the NCBI website. On the NCBI website, we will go to protein database. We will select protein here, and after that I will type the name of the sequence which we want to model. So here it is a list of uh, related sequences. So for example, we need to model this first sequence, HSP8 protein partial having 219 amino acids. So we will click on this, and after clicking on this it will open up the details of this protein. Then we will click on FASTA, and this will open up the amino acid sequence of the protein. Now we can just copy this sequence here, and now we will go to our desktop. Here we can create a new text document file, and in this text document file we will paste our FASTA sequence. So FASTA sequence had certain format, so but this format is not compatible with modular, so we need to make some changes. So what changes we need to make? We can find it in the tutorial for modular. So for this demo we are using modular 10.3. So I will open tutorial for modular 10.3. So I will go to this website, and here you can see here it is tutorial. I will click on the tutorial and then this basic modeling. So before uh, making the changes, I would we need to download this zip format files, so that uh, because it contains some in files which are required to create other input files using modular. So I have already downloaded this, so I will just go to downloads, and in downloads, uh, this is the folder which I have downloaded and extracted here. So from this folder we need to copy this pdb underscore 95.pr file, and we need to paste this file in C drive where our modular is installed. So I will open C drive and then go to program files and then modular 10.3, then bin, and here I will create new folder and I will name it modeling, and then I will paste that file here. Now we will be using this folder for our further files and for further operations. So I will minimize this, and now here we can see this is the format for the query sequence in for modular. In this, the starting the header code, this one is different from what we see in our FASTA sequence. So we need to copy uh, this this code, and we need to paste it in place of the header code of this FASTA sequence. So I have pasted it, and now we will we can see that at the end of the sequence there is a star. So that star is also required for FASTA query sequence, so I will put a star here. And now in this sequence, in the tutorial we can see they are using tvldh for the tutorial. They are using tvldh, but our sequence is different, so we have to we can give certain name to our sequence in place of tvldh. So I will open the faster the downloaded sequence, and here in place of tvldh we can give just we will replace all the tvldh wherever it is written tvldh with query, and then we'll replace all. So we can see the tvldh it is replaced with query. Now our query sequence file is ready. Now we will mm, go to file and save it as query.ali. That .ali extension is very important, so we need to save it as query.ali, and we will remove the text extension .txt extension. And first we will save this on desktop, because if we directly save it in the folder we created, it will ask for administrative privileges. So I will save it on desktop first. It's saved on desktop. I will close it. I will go to desktop, and here is the query.ali file. Now I will delete this previous new text document file, and this one I will query.li I will cut, and I will paste it in the folder which we have created in the bin folder earlier. So I will paste it here. Now it is pasted. Now we will go to the tutorial, and in the tutorial now we need to run a script which will create certain input files for the modeler. So this is the script. This complete is the script. Again we will do the same thing. We will copy this script, and then we will go to desktop. We will open new text document, and we will paste our script here. And now in this script also we need to make certain changes. In we can see in the script they have used tvldh here. So just we will tvldh.ali, but our file is query.ali, so we will replace this tvldh with query. So we will replace also that wherever it is tvldh it will be replaced with query. So no other changes are required, so we will just go to file and save as. Now we will save this on desktop, and we can give any name like script1, but we need to give the file extension as .py, script1.py. .py extension is very important for to make it readable to modeler. Now I will save it. I will close it, and we can see on the desktop this file script.1.py is created. I will delete this older file, and then I will cut this file, and now I will paste this in the folder, modeling folder, which we created. So now now we can see it is pasted here. Now we need to run this script to create other input files for modeler. So for running the script, we need to go to modeler. So I will open modeler, and one thing is very important: we need to run modeler as administrator so that we do not get any error. And so I will run it as administrator. So this is the modeler command line. Now we will, the folder, active folder is modeler 10.3. We will move to our folder which we created. So cd uh, cd space bin backslash uh, modeling. This is the folder we created. Now we have opened that folder here in modeler command line, and now we can type dir to see the files present in this folder. So we can see pdb underscore 95, which we downloaded from the tutorial, and this is query.ali, which we created, the script 1.py, which we created. So we need to run this script. So we will type the command to run this script, that is mod 10.3. 10.3 is the version of modeler we are using, space script1.py. Now we press enter, and the command will run. And after the command is run, we will see certain files are automatically created in the modeling folder. So we can see now this these three files have already are created, like script1.log, buildprofile.li, buildprofile.prf, and build pdb underscore 95.bin. These are the new files created which will be used by modeler later. So now our first step is done. So we will move to the next step, which is the selecting the template. So in selecting the template, first we need to run a protein blast in NCBI itself for our query sequence, and then we will get uh, certain sequences which have matching sequence identity. So we will select the top 3 to 4 template from those based on their score, and then we will download the pdb structure of those templates, and we will save those in the same folder which we created, and then we will run the script for selecting the best template. So I will show it now. So we'll go to the NCBI site again, and here we can see our sequence which we are modeling is already there. So I will click here on run blast. So after running blast, a page will open where it will ask for certain parameters. So here we will select the database as protein data bank proteins, because we need the pdb files of the templates. So I will click here blast now, and now it will take 2-3 seconds to give the results. And here now okay, we can see here that the top 3 to 4 sequences have, like, query cover is more than 84%, their percent identity is above 80%. So these are significant values. So we will select these top 4 structures. So now we need to download these 4 structures. So we will go to uh, the pdb website, that is rcsb.org, and then so it is rcsb.org I will open, and then we will type the pdb id, this 4po2 here. So here I will type 4po2 and I will select this. So it will open the page. For now we can go to this download files, and here we can click on pdb format, so that we will get the pdb format of this file. So now we will copy and paste this file to our folder. So before copying and before pasting it there, we can just write some other name, suppose temp1, template1, so for our convenience. And now we can just cut it and paste it in the folder we created in modular 10.3. So I will paste this here. Similarly, we will download other files and change their names and paste in this folder. So I have already downloaded the other two more sequences, and I have pasted them in the changed their name and pasted them in the modeling folder. These are temp1 already. I showed you how to download this. Then temp2 and temp3 I have downloaded and pasted it here. Now after this we need to go to the tutorial again, and from here we'll go to this topic 2, selecting a template, and this is the script for selecting the template. So I will copy this script, and again I will paste it on desktop. So in a new text document file, so I have pasted it here, and in this we will make changes accordingly. Here we can see they have given the name of the sequences which they have used in the tutorial. So this we will replace with our template sequences which with the names which we have given. So this I will replace with temp1, 1 BDM I will replace with temp2, 1 CIV I will replace with temp3, and 5 MDH I will replace with temp4. Now they have used more than uh, we are using 3 sequences only, so this temp4 is not required. So the rest of the sequences which they have used in the tutorial we can delete it. So we need to delete this. Okay, now this is done, so there are no more changes required. So I will save this file in py format. So I will save it on desktop. I will write it at this script 2.py and save it on desktop first. Now we can go to the desktop, and that script 2.py file is here. We can delete this text document file first, and then we will cut this script file and paste it in the modeling folder. Now the script is here, so we can now we need to run this script. We will go to modeler command line, and here okay, so now I will type the command for running the script, mod 10.3 space script 2.py. So now script 2 has run. So we will go to our modeling folder, and here we can see few more files are generated. So this script 2.log and this family file, this has been created by modeler. So now we need to go to the tutorial again, and now we will move to before that we should go to the modeling folder, and we can open this script 2.log file. We can open it with notepad, and at the end of this file we can see a comparison between the three templates which we used. So here the template 1 is the best template. This 2.0, it shows its crystallographic structure resolution. So we will select this template 1 from these three sequences for further operations. So now we can close this script file, and now we will go to the next step, that is aligning our sequence with the template. So this is the script for alignment of our query sequence with our template sequence. So the now I'll copy this script. Again we will paste it in a new text document file, and we will make required changes. So here we can see, here it is one BDM which they have used in tutorial, the sequence which they have used in tutorial. So this one BDM, this we will replace with our template sequence, that is temp1. So I'll replace all, and then here we can see they have, it is written as tvldh. So tvldh we will replace with our query sequence. So I will replace tvldh with query. So we have made the required changes. Now I will save this script file as in .py format. So I will save it as script3.py. Now we can see on the desktop the script3.py file is created. So I will just delete the text document file, cut this script file and paste it in the modeling folder. Now script3.py is pasted here. So again we will go to modeler to run the script 3. So here we can type same command, mod 10.3 space script3.py. So now the script 3 is has also run. So it will create some new files. Here we can see these top two files, .pap file and .ali file has been created after running the script 3. So now we will move to our final step. Again we will go to the tutorial. So in tutorial the final step is model building. So this is the script for the building the model based on the selected template. So I will copy this script again and paste it on desktop in a new text document file. And here again we need to do some changes. So here we will replace tvldh with query. It's already written here. So and this one BDM I will replace with temp1. So I'll type temp1 and then replace all. So we can see that it is replaced, tvldh is replaced with temp1. Now again we will save this script in .py format on desktop. So this is script4.py. Now we can close this and we can see on desktop script4.py file is created. So I will cut this file here and paste it in the modeling folder. Now the script 4 is .py is present here. So we will go to modeler command line again and we will type mod 10.3 space script4.py. So this script will uh, create the model. So while this script is running, we can see in our folder some new files are being created. So these are the new files being created, and and the models for our query structure based on the template are also getting written here. So this is the first model, query b triple nine triple zero one is the first model, and this is the second model. It will create few more models, and then we will analyze these models. So it will take around 5-10 seconds to create all the models. And in this we can also see script4.log file. This script4.log file, this we will use to see the dope score which is assigned to the model by modeler, and based on the dope score we will select the best model, and then we will evaluate it using certain online servers and tools. So we will go to modeler command line, and here we can see that the script 4 has run successfully. So now we will go to the modeling folder again, and then here we will open script4.log file in notepad. So here we can see at the last of this file all the models are given, and their mol pdf and dope score and ga341 score is given. So based on the dope score, the model with having the lowest dope score or the most negative dope score we will select. So we can see this model 4, this is having the lowest dope score, so we can now evaluate this model using online servers. So we will go to uh, we can use saves server which is created by NIH. And so I will open this server here. uh, so here we can choose the model which we want to evaluate. So I will choose, we have seen in the script4.log file that is model 4 has the maximum most lowest dope score. So we will select model 4 and open it here. Now it is selected here, and now we will run programs. So here we can evaluate our model. It is a combination of several evaluation checks. So we will evaluate for errata, we will evaluate for verify 3D, and we will evaluate for pro check. So we will start with errata. So now it will take few seconds. So overall quality factor for this model is 84.9462, which is very good and very significant. Now we will verify that with verified 3D. So it determines the compatibility of the atomic model with its own sequence. So we'll start this verify 3D check, and here we have to wait for 5-6 seconds, and it will give the results. So, and after that we will go for pro check evaluation. That pro check evaluation does the it checks the stereochemical quality of the protein structure by analyzing the geometry residue by residue. So this verify 3D is taking a little more time. We have to wait. See the results of verify 3D has come now. So it is showing that 99.54% of the residues have averaged 3D 1D score that is greater than or equal to zero point 0.2. So if 80% of the residues shows the score greater than zero point 0.2, that models is passed. So we can see our model is passed here. Now we can go to pro check and start the pro check evaluation. So in it will, this pro check will perform few, and I think six or eight evaluation, and out of this it will show how much are passed. And so here we can see it is showing out of 8 evaluation there are errors in 2 and 6 are passed, and there is no warning. So this is a good result for the model. So we can see the evaluation, our model, based on this evaluation we can say that our model our query sequence has been modeled correctly. So now we can do some more evaluations with other tool, that is VADAR. So this VADAR tool, uh, we can generate our Ramachandran plot here. So I will choose the our model 4 again here, and then I will click submit. So it will generate the Ramachandran plot and other statistics for our model. Here we can see uh, these are the plots it has created, and these four are the output files it has created. From here we can click on Ramachandran plot, and it will give the Ramachandran plot for our model. So we can see that most of the this Ramachandran plot, so that the most of the residues are in allowed regions. uh, and then again we can go to the output of the WADAR 1.8 results, and then here we can click on statistics. So this statistics will show the percentage of alpha helix, the percentage of beta sheets, and the coiled structure in our protein. So alpha helix is 40%, beta sheets 21%, and coiled is 38%. So, and other statistics of our protein model structure we can find out here. So based on this evaluation, we can say that our model is significant and correct. Now we can visualize our model in many visualization software, like uh, I will open this PyMol or Discovery Studio. So I will open PyMol now, and we will visualize our model in the PyMol. So so see, in the PyMol is opening. Okay, so in PyMol we can just go to file, open, and we can select our model from from the modeling folder. So this is the model 4. I will open here. So here we can see this is the model structure for our query sequence, and we can check its alignment with the template sequence. So we know that our we can go to the NCBI, and we know that it is model based on the template 1, which is 4PO2. So we can just in PyMol we can open this 4PO2 and download. It will download the structure of 4PO2 in PyMol. So here we can see it has downloaded the structure. So we can see here that our structure is nicely aligned with a segment of the template sequence, and the rest of the sequence in the template doesn't have identity with our protein or with our query sequence. Our query sequence was 219 amino acids only, so we can see those amino acids are aligned nicely with the amino acids of our template sequence. So, so this is all with modeler. We have performed all the steps, like preparing the query sequence, then selecting the template, then building the model, and the model evaluation. So this is all with basic modeling, and there are certain other things like advanced modeling, which will, but in this demo I will be showing only basic modeling. So That's all with the video. Thank you.
So I hope you have enjoyed the demo clip, and in the demo clip my student has shown you the different steps how you can be able to find the query sequence, how you can be able to use that query sequence to determine or select the templates, what are the different parameters you should use, and while he was showing you the demo, he has used the program which is called as modeler 10th version, right? And then he has shown you how to do alignment of query with the template so that you can be able to screen out which template you should use and which template you should avoid. And then he has shown you the molecular modeling of building the 3D models, and then ultimately the validation of the program with the service servers. So now you got the 3D models at right. So what we have discussed so far is we have discussed about the tertiary structures, how to determine the protein tertiary structure of the proteins, and we have discussed about the computational method, and we have also discussed about the experimental wet lab experiments. So in the wet lab experiment, we discuss about the X-ray crystallography and the NMR spectroscopy, whereas in the case of the non-experimental computational method, we have discussed about the homology modeling. Now, once we are done with the tertiary structures, you can also have the quaternary structures. So, if the multiple polypeptides are involved in the constitution of the protein, the tertiary structure of these different polypeptide chain come together to form the quaternary structure. Now, the question comes, under what conditions you can have the quaternary structures. So, and how you will know that the protein has the quaternary structures. So, if so, the question answer of this question is, if the multiple polypeptides are involved in the constitution of the protein, the tertiary structure of the different polypeptide chains are going to come together, and that's how it is actually going to give you the quaternary structure. Classical example is hemoglobin, where you're going to have the 2 alpha chain and 2 beta chains, right? So, it is actually a hetero tetramer, right, where you have the 2 alpha chains and 2 beta chains. So, it has a 4 chain, and that's how it is actually going to give you a quaternary structure. But the question is, how experimentally, when I give you a protein sequence or when I give you a protein, how you can be able to determine whether it also has the quaternary structure or not. So, there is a simple experiment what you can actually do to determine whether the quaternary structure is present in this particular protein or not. What you are going to do is, methods to determine the quaternary structure is, what you are going to do is you are going to determine the oligomeric status, and this methods anyway we are going to discuss in detail when we are going to discuss about the gel filtration chromatography and as well as the electrophoresis. Right. So, what you are going to do is you are going to take the protein and resolve that protein onto a gel filtration column in conjugation with the SDS page. And what will happen is that when you do that, it is actually going to give you the 2 molecular weight. It is going to give you a native molecular weight which you are going to get from the gel filtration column, and it is also going to give you the denatured molecular weight which you are going to get from the SDS page. Now, what you are going to do is you are going to determine the oligomeric status, right? And when you do the oligomeric status, what you can do is you can determine the NM, right? So, native molecular weight you can actually be able to divide by the denatured molecular weight. So, if this number, so you will get a number, right? So, if this number is 1, it is actually going to have, so if this number is going to be 1, right, you will not going to have, so no quaternary structure, right, because it only has one polypeptide chain. If this number is actually going to be 2 or more, right, then it is actually going to have the quaternary structure because it is going to have the multiple polypeptides. So if I think you will not be you would be more curious that how we have actually so I'm sure you will not be able to understand it very clearly because so far we have not discussed about the gel filtration chromatography, even the SGS page electrophoresis, but the idea is that you should determine the oligomeric status and then it is actually going to give you the idea whether the quaternary structure is present or not. If the oligomeric status is 1, then it is actually going to be a monomer, right? And if it is quaternary structure is 2 or more, then it is actually going to be oligomer, and oligomers are going to show you the quaternary structure. Now, irrespective of whether we use the molecular modeling, which means whether you use the homology modeling or whether you use the X-ray crystallography, you are going to use the NMR spectroscopy, you are going to get a 3D structure of a protein. So, this is the typical 3D structure of a protein where you will see that these are the helix, these are the beta sheets and they are arranged together, and what you see is these are the unstructured loops. So, these are the loops, these are the beta sheets and these are the helix, right. Now, when you have a protein structure, it actually requires a very extensive study with the help of the different types of softwares what you can use to determine many properties of this enzyme or many properties of this particular structure, right. So, we have prepared a very small demo clip to show you what are the different properties you can actually be able to study from the protein structures, and how you can be able to exploit that information for developing the substrate or the inhibitors, and how you can use that for studying the different types of interactions. So, this demo clip is actually going to be very useful for learning the how to analyze the protein structures. Hello everyone. So, in this particular demo, we are going to see how you can be able to analyze the protein structure, and so for demo purposes we have used a software which is called as Pymol, and Pymol educational version can be easily be downloaded from the Pymol's website. And then what you have to do is first you are going to load the molecule. So, for loading the molecule you just first click the file, open, and then you go to the respective directory. So, we will go to okay. So, I will load the molecule. So, I am loading a molecule which is called as protein PFD0975W. It is a malarial protein. So, once you load the protein, it is going to show you like this, and what you are going to do is, so what you see in this protein is that there is a ligand what is actually been present, right. And now our first objective would be that we would like to do the active site analysis, okay. So, for doing the active site analysis, what I will do is, I'll make it little beautiful. So, what I am going to do is, I am going to convert this into a cartoon model. That is the model what you normally see, and then we are going to see the primary structures, we are going to see the secondary structures, we are going to see the tertiary structures, and then ultimately I'm also going to show you how you can be able to do the analysis for the active site. So, for making a cartoon model of this particular protein structure, what you are going to do is you click the hide. So, you hide first the protein and then you are going to put the cartoon. Okay. So, moment you do that, it is actually going to give you the cartoon. Now, if you are interested and you want to make it like secondary structures, or you want to make little coloring to this structure so that it is easy for you to identify, what you can do is you go to this color tab, right,
and then you can be able to color everything by secondary structure elements, or you can do the molecule color by its choice, the color of your choice, right? So what I'm doing is I'm putting the by secondary structure. So I am choosing this, right? So where the helix is going to be colored as cyan, sheet is going to be colored as pink, and the loop is going to be colored as orange. Okay, so now this is what you're going to see. Okay. Now, if I want to study the primary structure, so if you recall, primary structure means the amino acid sequence. So, if you want to see the amino acid sequence of this particular protein, what you can do is you are going to click this S. You see, there are so many buttons here. So, if you click the S, there will be a window which is going to be pop up on the top, and that window is actually going to give you the idea about the amino acid sequence. Okay, and it actually is a very handy tool, because it is actually going to help you to select a portion of the protein which you are interested to zoom or something. Okay, so in this if you see the second primary structure, what you see is that different region is colored with a different color. So for example, the beta sheets are colored in a pink, right? So this is the beta sheet residues. Okay, so these are the residues which are responsible for making the beta sheets. So this is, see, all these beta sheets are being selected. And similarly, for helix, for example, this is the amino acids which are making the helix. So this is the helix what they are making actually, right? And so on. In some cases, you might have to select, for example, if you want to use this protein for, or if you want to use this enzyme for designing the drugs, right? So in that cases, you might have to study some region of the protein, and then you have to select. So in that is also very handy, right? If you just like select, for example, I have selected the amino acids from 150, right, to 200, for example. So I will select up to 200, right? And then what I'll do is I want to color that region in a separate color. Okay, so I have selected it, right? So this two button, right? One is for protein, the other one is select. So in the select, I will go and I'll say, okay, this is the color I want to choose. So I'll choose like magenta, or I will choose like some other color which is not there. Okay, so I will use the yellow. So this is the portion, you see, this portion is got labeled as yellow color. So that actually is going to help you to identify, okay, this is the portion which is responsible for catalyzing the reactions or interacting with the substrate and so on. Now, as far as the, so this is all about the primary structures. If you scroll this at the end, it will actually going to show you. So this is actually it's showing the PDB. So it's also going to show you that there are manganese, two molecules of manganese which are present in the protein structure. And then it is having the protein bound phosphate, and it also has the ATP at the end. Okay, so it also has the protein bound ATP, because this protein is a kinase. So it is actually going to have the active site bound ATP as well. Now once we have gone through with the primary structure, we can just click this, and then it's actually going to disappear the tab. And then you can also study the secondary structures. So for example, this is the helix, right? So what you see, right? Beautiful cyan colored helix, and then these you also have the beta sheets, so pink color beta sheets, what you see, and see all the two strands are so this is actually anti-parallel beta sheets. So one sheet is going in this direction, the other one is going in this direction. So this is a anti-parallel beta sheets, whereas what you see, this is also a anti-parallel beta sheets. One sheet is going in this direction, the other sheet is going in this direction. Whereas if you see very carefully, the this sheet, this sheet, or this sheet and this sheet is actually in a parallel mode. Okay, so they are arranged in a parallel mode. For example, these also are anti-parallel beta sheets. And what you see, right, all these secondary structures, right, alpha helix or beta sheets are connected by the loop structure, and you see how the loops are unstructured, right? And because this is a modeled structure, what you see is basically having the lot of unstructured region, because this is the structure, this is the region where the protein does not have any kind of homology with the existing structure. So if I want to improve this, I probably could have to do ab initio molecular modeling. So this is all about this. Okay, now if I want to do the active site analysis, what I'll do is I'll first put these substrate, right? So I will put the molecule, and so I will put the ligand, and I'll show that as sticks, right? So this is I have selected. Now you can see, right, this is the ATP, which is protein bound ATP. So this is the adenine ring, this is the sugar, and this is the phosphate. If I want, I can actually be able to color this as per the elements. So I can color it as per the element. So in that case, it is actually going to show me the phosphate, and the so wherever it has the negative charge, it is actually going to show me the red color, and wherever it has the polar groups or positively charged, it is actually going to give me the blue color actually. Now if I want to study more about this, so there are many ways in which I can be able to study the active site. For example, if I want to know what are the residues which are interacting with ATP, so what I'll do is I'll select the ATP. So you have to do nothing, you have to just click this molecule, either you click here or you click in this tab, and then what I'll do is I'll select, and I will do a right click. When I do a right click, I will say I want to see what are the different molecules are interacting with this molecule. So what I'll do is I'll go to the action, and then I will say I want to see what are the atoms are present within the 4 angstrom or 5 angstrom. So when I do that, it is actually going to select all the atoms of the proteins which are going to be present in the 5 angstrom radius, because ideally what happen is that when you are actually looking for the possible interactors, you always have to select like that. And then what I'll do is I'll go to the label, okay, and then I'll say I will label the residues. So what you see here is it has labeled all the residues which are within the 5 angstrom of this particular molecule, and that's how it is going to give you that, okay, these are the molecules which are probably be interacting. Now ultimately what you can do is you can do a distance matrix, and you can actually be able to open this molecule in other softwares to measure the distances. Apart from this, you can also be able to check the you can also be able to make the cavities. Okay, so you can also study how the grooves are present in this particular protein. So for that, what you can do is you can just go to here, and you say I want to see the surface. Okay, so what you are going to do if you want to see the charge distribution on the protein structure is you're going to see this, you're going to go to this action button. Okay, so click the action button, and then you say I want to generate the electrostatic charge distribution under the vacuum. Okay, and then you select this vacuum, and then you are going to select this. So it is actually going to calculate, and very soon it is actually going to show you the color. So this is the charge distribution what you see, and wherever you see the red color, that is actually the negative side, and wherever you see the blue, it is actually going to be positive side. Okay, now if I show you again the molecule, right, so if I show you the molecule again, right, so this is the ATP sitting here, right, and if I show you that ATP again, you will see that how nicely the ATP is being placed, and okay, so this is what you see, right, so ATP is placed, and wherever you have the blue, actually that is the positive, and wherever you have the red, that is the negative, and wherever you see this white, that is either the neutral or the hydrophobic regions of the proteins. Now this is all about what you can actually be able to analyze on the protein structure. One more thing which you can also do is you can actually be able to superimpose the two structures. Okay, so how to do that? what first thing what you have to do is you have to load the molecules. So for example, I have loaded two molecules like 1ZAO and this molecule, right? So if I load the 1ZAO and this molecule, I'm going to see the two structures, right? 1ZAO and this one, right? So this is the other one, right? And what I can do is I can just do the alignment. So I can do is I will click the action button, then I do the alignment, and then I'll say to molecule, and then you select. Okay, the moment you do that, okay, it is actually going to align onto the other molecule. To make it very simple, let me just remove all these molecules so that uh, so that it will be easy actually. So if I open ZO, one ZAO and protein, right, you're going to see that, okay, these are the two protein structures, right? So one is the 1ZAO, the other one is the PFD0975W. And if I want to superimpose this to this, what I'll do is I'll go here, I'll go to the align, I'll go to the two molecule, and then I'll select the other one, okay, and it is actually going to show me the alignment, okay. Uh, and what is the advantage of making an alignment? The advantage of making an alignment is that it is actually going to show you the contrasting features. So this is all about the demo on which so that you can be able to do the different types of analysis onto the protein structure. Thank you.
So I hope you might have liked the demo clip and you will understand the potential of the protein structures and how the protein structure is actually very complicated, and it requires a very detailed study to understand the function of each and every portion of the proteins. So in that demo clip we have shown you how to determine the charges, this charge distribution on the protein, how you can be able to explore the different cavities what are present in the protein structures, and so on. So what we have discussed in this particular lecture, we have discussed about the primary structures, we have discussed about the secondary structures, we have discussed about the tertiary structures, and we have also discussed about the quaternary structure. While we were discussing about the primary structures, we have discussed about how you can be able to determine the primary structures and what are the different types of amino acids are present. Amino acids are actually having a general structures where you have the amino groups, carboxyl groups attached to a central carbon which is called as C alpha, and then it is attached to the different types of R groups, and depending on the R groups you can be having the amino acids of different types, whether it is the polar amino acids, non-polar amino acids, hydrophobic amino acids, charged amino acids like the negatively charged amino acids, positively charged amino acids, and so on. And then we have also discussed about how you can be able to determine the primary structures with the help of the different types of protein sequencing methods. Now, once you have determined the primary structures, you can actually be able to understand the secondary structures. So, because these primary structures are only going to direct how the protein is actually going to fold, and that's how it is actually going to give you the secondary structures. So, within the secondary structure, we have discussed about the alpha helices, beta sheets, and we have also discussed about the loops, right? I already suggested you when we were discussing about the secondary structure that you should actually go through with some of the standard biochemistry books like the Leininger, White and White, or Stryer. So this is all about the different details of the protein structures and how you can be able to use the different techniques to determine the protein structures. So with this, I would like to conclude my lecture here. Thank you.